Okay, so we have an exam on Tuesday. Whoops, it's going. Tuesday. So that's November 1st. Can't believe it's November 1st coming up so soon. It's going to be the same format as the last exam, so it's 100 points. So 40 points are going to be 40 multiple choice questions, so that's going to make up 80 points. The remaining 20 points are going to be things like matching, short answer, label the diagram, that sort of thing, right? Similar to what we had before. Study materials, this time you have 3 chapters, so I think it's 1 less chapter than last time, but yeah, because the last time was 1 through 4, right? But these are kind of difficult, probably more difficult chapters than you had previously. So what can you study? You could look at the lecture slides, which are posted on Blackboard. You can go to the Connect online quizzes. I think you should be able to see them, even after you finish them, right? You should be able to use them for study purposes. You can go back to the homework, review those questions. At the end of each chapter, each PowerPoint for each chapter, there was a list of questions, right? 10, 12 questions, that sort of thing you could take. Again, students that have done well typically will take those seriously and go ahead and answer out those questions at the end of the lecture slides. Here's some. Okay, go ahead.
Is there a way to make chapter 8 the quiz like doable before the due date? Because like you can't do it until the 31st, because that's when we like you can't review our results.
Oh. Yeah, I don't know if there's a way to do that unless I move it up for everyone. So email me about that. Let me see if I can do something, but off the top of my head, I don't know how I can change that. So you're saying that, yeah, you're not able to see it until the deadline is passed. Email me, I'll try and think about it. I really can't think of anything on how to solve that problem, but right now, but I can think about it. Okay, so here's some, obviously you should look at all those questions at the end. Here's some kind of key ones to think about. Where does the substrate bind to an enzyme molecule? How does the induced fit model help an enzyme to bind to its substrate? Describe the factors affecting enzymatic reactions. We did some of these in lab, right? We did things like temperature. Did we do pH? I can't remember now. Yeah, we did pH. Yeah. So basically, these things affect the environment around the enzyme, right? So that changes the activity. So that's something to review. What is a redox reaction? So again, so your book goes through this. We went through it in class. How does a chemical substance become oxidized or reduced in a redox reaction? We're gonna, and I have some slides coming up in it with some pictures. So we'll talk about this stuff again in a second. Both stellar respiration, photosynthesis are examples of redox reactions. Justify that statement. What is the difference between substrate level and oxidative phosphorylation? We'll talk about. I've got some slides coming up, so we'll talk about it there. So these are different ways of making ATP synthesis. Which way cells make more ATP? Which way make cells? Some of this is not very grammatical, is it? Which way cells make more ATP molecules? Oh, which way do cells make more ATP during cellular respiration, right? So obviously it's oxidative phosphorylation, right? So when you have oxygen available, you can break down glucose completely, right, into CO2. 5, distinguish between fermentation and cellular respiration, for example. So the term cellular respiration is a little bit general, but what we're saying here is we're really talking about aerobic respiration. So cellular aerobic respiration. Fermentation, of course, doesn't require oxygen. Again, we'll talk about that in a second. Distinguish between lactic acid fermentation and alcohol fermentation. Which kind do we do? Which kind of fermentation do humans do in our bodies? Yeah, we do the lactic acid fermentation. Notably, we do it in various parts of our body, but probably the most notable place is within skeletal muscle, because you need a lot of ATP very quickly when you're doing intense exercise. Like think of someone doing judo, powerlifting, sprinting, that sort of thing. What is a photosystem? We talked about that. So again, I'll have some slides coming up. How does it work? What is chemiosmosis? How is ATP synthesized during photosynthesis? And 9, what are the roles of ATP and NADPH in the Kalman cycle? How do they recycle? How are they recycled? Okay, so I think some of these you probably already answered some of these ideas, but let's look at this first question here. How do enzymes work? So the substrate. So remember that when the substrate is the same thing as a reactant, right? Those are sort of synonymous, but when we talk about enzymes, we often use the word substrate, okay? Because the enzyme is acting on the substrate. The substrate binds to the active site of an enzyme, which is typically kind of like exactly as shown here, it's a little bit of a groove or a notch in the enzyme. The substrate can fit in there. And we have the induced, so that forms then an enzyme substrate complex, and we have the induced fit model. This means that the substrate fits in the active site, but not perfectly. But then we've discussed the idea that these proteins, part of the sort of the magic of how they work is that they don't have rigid structures because they have hydrogen bonds, ionic bonds, hydrophobic, hydrophilic interactions that determine the three-dimensional shape. They're able to do a shape change. The fancy term in biochemistry for shape change is conformational change. They'll change their form to fit the substrate. And then what that does is, what's the first point of that? Well, then that can put sort of mechanical stresses on the substrate. And this can actually help. Say this is we're going to break the substrate in half. Well, that stresses the substrate once we do that. And it helps the reaction to go forward. So the active site undergoes a slight change in shape to accommodate the substrate. This is called the induced fit. The change in shape facilitates the reaction by achieving its optimal fit. And as again, it can actually put some mechanical stresses on the substrate. The active site can also, imagine we're putting two substrates together, so we're doing a synthesis reaction. The active site can also make sure that the parts of the two molecules that are going to be coming together to make a bigger new molecule are actually oriented correctly, that sort of thing, right? The change in shape facilitates reaction by achieving optimal fit. After the reaction is completed, products are released. So the enzyme is a catalyst. It helps increase the rate of reaction, but it's not used up in the reaction, right? That's the definition of a catalyst, and an enzyme is a catalyst. All right. What are some of the factors that affect enzymatic reaction? The rate of enzymatic reaction may be influenced by substrate concentration, right? So as you increase the substrate concentration, initially, when you have a low substrate concentration, let's say you double the substrate concentration, then you'll double the reaction rate. But eventually what happens, you guys remember this? Eventually what happens, so RxN stands for reaction. So here's reaction rate on the y-axis. On this x-axis, we have substrate concentration. So we have, imagine we have a beaker. We have a certain amount of enzyme, so we have a certain concentration of enzyme. So initially, it's very much a linear relationship, as you say. So if you double the substrate concentration, the reaction rate will double. But eventually, you get to this sort of thing, right? Where it sort of levels out. In fact, it becomes completely flat. What's happening there? Does anyone remember? Why is there a maximal reaction rate? You have a given concentration of enzyme, the substrate concentration continues to increase, which is shown here, right? So say we go from here to here, we've doubled the substrate concentration, but it really hasn't changed. What's happening there? Oh, go ahead.
You can only react with so much substrate at one time.
Yeah.
So if you just have more and more substrate, you can't react with something.
Perfect, right, exactly. So you can think of this here, I'll draw it like a Pac-Man. There's the enzyme, right? So let's imagine you have, early on, if you have a very low, let's try 4 of these. If you have a low concentration of substrate, so the substrate will be a little triangle thing. Okay, as I double the substrate concentration, so there's more of these, because they have to actually literally bump into each other. Okay, if I doubled this concentration, now I've doubled it, because there's twice as many going to the active site. It goes in for a little moment, for the active site, and then comes back out again. But at some point, when you really increase the substrate concentration, guys, remember we talked about this? Then you get to the point where they call it saturation, because the instant one comes out and it's changed into something else, it's changed into a star, say, a new one comes in, right? So here's a star. So you reach a limit, right? Because the enzymatic reaction, although it's incredibly fast, still takes some time, right? So that's why you get this to the saturation point. So that's the relationship between substrate concentration. Always looks like this. And this is in a very important curve, not just for enzymes, but also for transport of molecules across membranes and things like that. We talked about that also. So for the example, within your kidneys, the transport of glucose also follows this rule. So with people that are diabetic, well, first of all, you shouldn't have any glucose in your urine, right? You have glucose in your blood. It ends up in the pre-urine, the stuff called filtrate, but then your body thinks it understands that glucose is very valuable, so it takes the glucose back up from the pre-urine and it goes back into your blood. But in someone who's diabetic, well, certain types of diabetes, they have very high concentrations of glucose in their blood. And what they end up doing is saturating all the glucose transporters within the kidneys. And that's why they end up having glucose in their blood and then their urine, excuse me. Temperature and pH. Do people remember why would temperature, what are the effects of temperature? Let's do temperature first. What happens with temperature? Go ahead. Yeah, so let's do temperature. So here's a new plot. Eventually it'll denature, right? So here's our reaction rate. Here's temperature. And you're typically going to get this sort of a curve. It's going to vary depending on the enzyme and so forth, but there'll be an optimal temperature. So for us, a lot of our enzymes will work really well around roughly 37. You get kind of a peak right around there. Now, early on, what's happening is you increase the temperature. Well, if you have a very low temperature, this means that the molecule, there's less thermal energy, right? So there's a thing called activation energy you have to get over. It's sort of a bump. And so with low temperatures, even though the enzymes help reduce activation energy, there's less sort of thermal energy for the molecules to bump into each other, first of all. And then secondly, when they do bump into each other, they have less energy, kinetic energy. So it's harder for the reaction to go forward. But so this is going to increase, but eventually you start to see this decrease. And as Chloe said, what happens is your protein starts to sort of unravel a bit, right? So it starts to lose its three-dimensional shape, right? And then your reaction rate will go down. Now pH is going to be similar, but for slightly different reasons. So here you have most, like 7.4 is the pH of our blood. So it really depends, but a lot of our enzymes in our body fairly have, the best reaction rate roughly around 7.4, 7, somewhere neutral like that. Now there's an exception if you have enzymes that are active in your stomach. They might have a peak around 4, okay? Which makes sense because your stomach is very acidic, right? And the same thing happens with pH. There's similar reasons. Like basically what's happening is if you go away from this optimal pH for a particular enzyme, then you essentially get denaturing of the protein in either direction, right? Because what does pH represent? It represents the hydrogen ion concentration. And so the hydrogen ions can interact, particularly with things that are polar side chains on amino acids, as well as charged amino side chains on amino acids, and therefore affect the three-dimensional shape of the enzyme. Enzyme activation. Enzyme activation. So sometimes your cells will make enzymes, but they're not immediately active. A good example of this would be within your stomach. You have a enzyme that's involved in breaking down protein, and you don't want to break down your stomach wall, right? because it's also made out of protein. So what you do is, what your body does is it releases an inactive form of the enzyme, and then it gets activated within the stomach itself. So it can then, digest your meal. So what can you do? Well, there's different ways that you can cleave a part of an enzyme, so that can happen. Another thing is you can add or remove phosphate groups, right? Phosphate is PO4. You can also have an association between the enzyme and a protein or some sort of cofactor. A cofactor might be something like a metal or something like that. Okay, enzyme inhibition, when a product is abundant, it binds to an enzyme's active. Okay. So do you guys remember this idea? It's a type of inhibition. So you have these metabolic chains where you have enzyme 1. We're going to take a product. We're going to take something like this. Here's the substrate. Enzyme 1 will make a triangle. And then we have enzyme 2, which will turn it into a hexagon, that sort of thing, and so forth, right? And then what can happen is this last product here, and it could be a much longer chain, will come back over here, and it can actually fit into the well, here's the enzyme over here. I'll draw a picture of the enzyme. Here's the enzyme. So this product will fit into this active site. And what it does here is it will block the activity of enzyme 1. You guys remember this? And if it does that, then it stops the production of this along this whole chain. It could be much longer, right? It could be like eight different reactions or something like that. So that is a very classic kind of enzyme inhibition. It's essentially inhibition by the end product. So that happens a lot within your cells. When the product is abundant, it binds to the enzyme active site and blocks further production. Now, there's another type that's also of inhibition that's actually very found everywhere in your body, also within your cells, is allosteric inhibition. This is where you have a product that binds to a site other than the active site and changes the shape of the active site. So I can modify this slightly and say, okay, here's my enzyme, here's the active site. But this hexagon will bind over here. There's a little spot right on the other side of the protein. So it's allo means other, and steros means solid object. It's sort of like a three-dimensional object, steros. So this is a reference to the fact that the regulatory site of the allosteric protein is physically distinct from the active site. So what happens is binding at a distant site on the protein will cause a conformational change to the active site. So now it kind of closes up, right? So it won't accept, it can't accept the substrate anymore, right? The substrate will not go in, right? So we can indicate that by, this is textbook ease. They show this sort of thing with the arrow where it tries to go in, but it can't, right? So that's allosteric inhibition. Do we have also, many of the enzymes in your body require cofactors or coenzymes. So a cofactor is going to be an inorganic ion. So it could be, for example, something like iron, right? So hemoglobin requires a cofactor. That's for transporting oxygen within your body, within your blood. So the cofactor there would be iron. But you can have a coenzyme, which is a non-protein organic molecule that assists in reaction. So an example of a coenzyme we've seen is NAD, right? And it picks up electrons, right? Picks up 2 electrons and it becomes NADH. Well, 2 electrons plus a hydrogen, right? So that's a good example of a coenzyme. It acts as a kind of electron shuttle. So without it, the enzymes wouldn't work, right? So they need these sort of helpers, right? So you can think of cofactors and coenzymes as being helpers. Cofactors are typically inorganic ions, often metals, but not always, right? And then the coenzymes are non-protein organic molecules. Okay. All right, let's go on. Chemical reactions that transfer electrons between reactants are called oxidation reduction reactions or redox reactions. In oxidation, a substance loses electrons, right? It's said to be oxidized. And that makes sense because oxygen is really electronegative. It really wants electrons. So it's often the substance that's receiving the electrons, but not always, right? The electron donor is called the reducing agent. So it gives up electrons to the other molecule. And therefore it reduces the other things. That's why it's called the reducing agent. In reduction, a substance gains electrons or is reduced. So why do we say it's reduced? Well, it's gaining electrons. So the sort of handy mnemonic to help you think of that, at least for me, is that electrons are negative. So if you add something that's negative, you're going to get something that's going to get, you're going to get more negative, right? Their numbers will go down, right? It will become reduced. So if you have a molecule that's an acceptor of electrons, it's called an oxidizing agent. So again, oxygen likes to accept electrons, so it's a good oxidizing agent. The term oxidation is used even when oxygen is not involved. Here's an example from cellular respiration. So here we have a carbohydrate, it could be glucose. We do have unequal sharing of electrons because the oxygen shares, but we also have a lot of equal sharing of electrons between carbon and hydrogen. So if you pay attention to carbon, you'll see that now when carbon becomes oxidized, it doesn't completely lose the electrons, but it does somewhat lose electrons because now it's unequally sharing in CO2, which looks like this. The electrons actually like oxygen so much that even though they're sort of buzzing around the carbon and the high oxygens, they're going to spend more time near the oxygen end. So you could indicate that with a sort of a delta. It's a partial negative charge near the oxygen. So that is an oxidation. Here, O2 becomes reduced when it combines with electrons, when it gains electrons plus hydrogen, right? So what's happening here is in oxygen, we have essentially a battle. Is it a double bond? Some of these chemistry people, is it a double bond between oxygen? Yeah, I think so. Okay. And then here's H2O, you have single bonds. So remember that we have these partial positive charges. That's a delta, that's a Greek letter delta. Partial positive charges on the hydrogen, partial negative charge on the oxygen. And what that means is these are polar bonds, meaning that the electrons are spending more time near the oxygen than near the hydrogen. So the oxygen is being reduced because now the electrons are really spending more, they're closer to the oxygen atom than they were here, where they're the electrons are being equally shared between the two oxygen atoms. And then it's a little confusing, but this thing here is a reducing agent because it gives up electrons to the oxygen. Oxygen is an oxidizing agent because it accepts electrons from the carbohydrate. All right, then we also looked at substrate level phosphorylation versus oxidative phosphorylation. So which of these two pathways is the most ancient? Which one arose earlier in life? Substrate level or oxidative phosphorylation? Substrate level is much more ancient. So bacteria, archaea, basically every living organism does substrate level phosphorylation. They all do glycolysis. Maybe pretty much it's the same as within us, with if there's some slight variations, they're very slight. So what is substrate level phosphorylation? It means that you have an enzyme, you have an enzyme that can essentially transfer phosphate groups. So here we have an atom, it's a 3-carbon organic molecule. It has 2 phosphate groups, which is PO4. It can move this enzyme, with the help of this enzyme, we can remove one phosphate group, put it onto adenosine diphosphate, which becomes adenosine triphosphate. And then this is a good energy source within our cell. And then you can see this molecule comes out having one less phosphate group. Okay. So we're moving a high energy phosphate from one molecule and passing it to ADP to form ATP. So your cells do that all the time, right? During glycolysis and also during, so during glycolysis. Now, what your cells have and all eukaryotic cells have is another system for making ATP, which is oxidative phosphorylation. This is a process in which ATP is formed as a result of the transfer of electrons from NADH or FADH2 to O2 by a series of electron carriers and electron transport chain. You guys remember this? This is the so we've got the matrix in here, right? Within the mitochondria. What's this space outside here? What's this membrane here? Remember the mitochondria, it's a double membrane structure. There's an inner and outer membrane. Which membrane are we depicting here? Which one? Yeah, the Christa singular, Christi plural, or the inner membrane, exactly right. So this is the inner membrane, and remember how it's got all these unfolding? Because this is where the electron transport chain is, and it's got all these different proteins and things like that in it. So it's just absolutely packed with proteins. And this is the matrix in the middle, right? So this means if this is the inner mitochondrial membrane, there's another membrane out here. So this space here is it's between two membranes. So what is it called? Intermembrane space, right? Inter means between, so it's the between membrane space, the intermembrane space. So this is essentially where all this stuff is happening. So remember, within there was a preparatory step and the citric acid cycle. Those occur within the matrix, right? Of the mitochondria. And is it hot in here? I think I'm gonna battle with someone else. Yeah, someone I was pretty cool. Someone else always puts all right. The thing in this battle between people who like it hot and people who like it cold is that as a teacher, I'm not allowed to just come in flip flops and shorts. I mean, that would not be professional, right? So, whereas if you're cold, I mean, okay. You see my point there? You can just put a sweater on or a sweatshirt. I mean, I literally cannot come in. I would love to just come in with shorts, but it doesn't make any sense to me. All right, so you don't want too many. All right, so all right, it's hot. It's hot because I'm also talking, so it's really hot. Sorry about that. So we're harvesting stuff. Remember that? We've got the citric acid, the preparatory stuff, citric acid cycle. Remember, it's harvesting electrons. So we have the NAD becoming NADH. What was the other? The FAD picks up electrons, becomes FADH2. So those are like essentially little shuttles, right? They're moving electrons to the electron transport chain. NADH donates those electrons to go to the electron transport chain. And then that energy, as electrons fall down in terms of energy, is used to pump hydrogen ions. What's the direction? Well, it pumps the hydrogen ions into the intermembrane space. You guys remember that? Now, the FADH comes in a little bit later. So for the electrons that you put in with NADH, you're going to get more hydrogen ions pumped across than with FADH, even though it's 2 electrons, just because of the FADH coming in a little bit later. So for example, in this picture here, the NADH comes in here, you're going to get 1, 2, 3 electrons pumped, whereas with the FADH2, you're just going to get 2 electrons pumped. Does that make sense? And I don't care about, some people care about keeping track of all the numbers. I don't care, but you should know that you will get less essentially energy out of the FADH2 compared to the NADH. Okay? All right, so then we do that. We pump all these things across. Finally, the electrons have to go somewhere. At this point, you guys know this, right? It goes to the final electron receptor, which is oxygen. We have hydrogen ions in the matrix, so you pick up electron. The oxygen picks up, O2 picks up electrons plus hydrogen becomes H2O. Your cells are making water all the time. It's actually in a sense a kind of a waste product, but it's useful to have water too. Now, we generate an electrochemical gradient, also called proton motor force, where we have a lot of hydrogen ions in the intermembrane space and fewer in the, so there's a difference in concentration between the intermembrane space and the matrix, such that they're going to want to move down their concentration gradient. There's also an electrical gradient because the hydrogen ions are positive. So the inside of the matrix becomes negative with respect to the intermembrane space. Therefore, there's a very strong force for hydrogen ions to move down, simply downhill, right? And they're going to move through the ATP synthase, which generates ATP from ADP and inorganic phosphate. And eventually they're showing here that the ATP has to come out and then that's used for cellular work, okay? So our cells do this. So this is a very advanced system. And basically with it, you can break down something like glucose completely, right? You just end up with CO2 and a lot of energy. So the energy yield is really high. Again, I don't care about numbers as much as some other teachers, but it's in the 30s, right? Like 32 to 36, 38, something like that, some number like that per glucose molecule, right? Whereas if you weren't able to do this, the net output with per molecule of glucose to substrate level phosphorylation and glycolysis is only 2 molecules of ATP. Yeah, here's the numbers right here. If oxygen is present, glucose will be completely oxidized and you get something like 36 to 38. Now, this is the standard sort of textbook number. Some other calculations come out slightly lower because if you take into account the fact you actually have to shuttle things and that costs energy and so forth, you get actually slightly lower numbers. But that's the sort of standard textbook numbers. You'll see if oxygen is absent, cells may utilize anaerobic pathways for pyruvate is reduced to lactate. So that happens in our cells. And then in other organisms such as yeast, we did this in lab, you get alcohol and carbon dioxide. Okay. In either case, they're called fermentation. So we have lactic acid fermentation and alcohol fermentation. So lactic acid fermentation is also used in a lot of the product, a lot of food products such as cheese, yogurt. Right? That's lactic acid fermentation. Fermentation begins with glycolysis to produce 2 ATP, but skips the citric acid cycle electron transport chain. The entire process of fermentation takes place in the cytoplasm, results in the net gain of 2 ATP per glucose molecule. So can you survive just on lactic acid fermentation as a human? No. You can. It's good for like Usain Bolt when he runs the 100 meters in less than 10 seconds, that sort of thing, right? Because he's basically doing a lot of lactic acid fermentation in his muscles, but you can't really survive. It's not enough to sustain your life. It's good for short bursts of energy. In human muscles, lactate or lactic acid is the product. So remember, we talked about this. Lactic acid is the real product, but of course, it can donate its hydrogen ion, so it becomes the ionic form is lactate that you would actually find in your cells. First, glucose is oxidized to pyruvate. So glycolysis. Okay, here's our glucose. It's a 6-carbon glucose. We go through glycolysis, and we're simplifying everything a lot, but we're getting out our 2 ATP. Remember, we actually harvest some electrons. So NAD, we have 2 NAD. They become 2 NADH, and they've gained, each of these have gained 2 electrons. We split this, and there's a lot of steps here that are being glossed over, but you have then at the end pyruvate, each of which is a 3-carbon organic molecule. Now, one of the issues is that first of all, we're going to build up with pyruvate, but the second thing that's probably more important is in order for this reaction to go forward, you have to have NAD, right? So glycolysis will not occur without NAD. So how can we regenerate NAD? Well, NADH donates electrons to pyruvate, and you end up with lactate. Okay, so you end up with 2 lactate molecules. So of course, lactic acid is slightly acidic, so it has some effects within the muscle cell. But then we regenerate the NAD, and then we can keep generating more energy, okay, in the form of ATP through substrate level phosphorylation. So in yeast, pyruvate is reduced to ethyl alcohol, which is also called ethanol by fermentation. So that occurs every time you make bread, right? You make CO2 as well as ethyl alcohol. And you can't get drunk because when you bake the bread, you just lose the alcohol. Photosystems, the pigments that act like an antenna for gathering solar energy. Light energy, the photon is first captured by one of the chlorophyll pigment molecules and is then passed along to the other pigment molecules until it excites the reaction at the central, the reaction center chlorophyll. So you have different chlorophyll. Some are here at the reaction center. So we sort of funnel all these photons to here. We then excite the electrons here. The electrons are actually able to leave the atom altogether and go to the electron acceptor. But we have to we would run out of electrons pretty quickly. So where do we get the electrons? Well, we have hydrolysis, being breaking apart of water. So we get oxygen, hydrogen ions, and electrons. And that's how we keep feeding electrons into this photosystem. Okay, so which photosystem is this, guys? Photosystem 1 or 2 that's being depicted here? 2, right? It's the first one. Unfortunately, it's called 2, right? The first one that occurs. All right. So light energy in forms of photons is first captured by one of the chlorophyll molecules and past the other molecules, excites the reaction center chlorophyll. Upon excitation, the reaction center releases an excited electron, which is then transferred to an electron acceptor. The excited electron that is lost is replaced by an electron donor, so it comes from the water molecule. Okay. So this is showing you the light reactions here. We talked about that this all occurs within the thylakoid membrane, right? So inside of the thylakoid membrane, we have the thylakoid space. What's the name of the clear fluid that's outside of the thylakoid? Stroma, exactly. So what's happening here? Well, photosystem 2, we get the light coming in. We've hydrolysis where we break the water apart. The electrons enter into an electron transport chain. You can see here there's an electron, there's a proton pump. So protons are being pumped into this thylakoid space. There are also protons that are generated by the fact that the water molecules split, right? So we're increasing the hydrogen ion concentration inside the thylakoid space relative to the stroma. Now, the electrons actually end up going downhill in terms of energy, but then they get boosted again in photosystem 1. And then in the non-cyclic pathway, which is the only one we've discussed, it's the only one you're responsible for. There's a cyclic pathway, but we didn't get into it. But in photosystem 1, we have, again, this energy, you boost the electrons are passed, and 2 NADP, right? And with the aid of this enzyme NADP reductase, together with some hydrogen ions, you then have NADPH. So that's going to be one of these things where you picked up 2 electrons and we're able to use those electrons in the Calvin cycle. Now we've built up an electrochemical gradient, or proton motive force, where the protons now are much higher in concentration inside the thylakoid space compared to the stroma. There's also buildup of positive charges in the thylakoid space relative to the stroma, which would be negative with respect to the thylakoid space. So the hydrogen ions really want to go downhill, and they're going to go through the ATP synthase, and that's movement of the hydrogen ions downhill down this gradient will lead to the production of ATP from ADP and inorganic phosphate. And this is called chemiosmosis. Okay. Why is photosystem 1 over here? Why does it come after photosystem 2? Anyone remember? It seems illogical. What's the reason? What? Yeah. So they discovered this. This is historically what they did. They discovered this one first, right? And then they discovered this one. So it's a little unfortunate, but you can't really go back in time and change everything. So we talked about this before, right? So the light reactions, you have water coming up from the roots of the plants. The water is split because we need those electrons. and we get oxygen coming out, which is used by the plant as well as by us. We generate ATP, as I just showed you. We also, the NADP picks up 2 electrons. And those electrons are going to be used for reduction over here within the Kelvin cycle. ATP is used for energy. And then we go into the Kelvin cycle. The Kelvin cycle will take CO2 as a raw material, as a carbon source. And what are the 3 steps it's going to do? Does anyone remember? There were 3 basic steps. First one is we have carbon in an inorganic form. CO2 is inorganic. Right, it adds it to an organic molecule, right? And that's called carbon fixation, right? Right. So you need to first attach the carbon to an organic molecule. That's carbon fixation. What's the next step? The Kelvin cycle. That's a reduction, right? Yeah. So then we use the electron from NADPH plus with some energy from ATP to do the reduction reaction. So that essentially means we're adding electrons to the carbon, and along with electrons comes some hydrogen. So we're essentially forming the carbohydrate, which is a ratio of 1 carbon to 2 hydrogens to 1 oxygen. And there's a last step. What was the third general step for Kelvin reaction? The Kelvin cycle. Go ahead.
It's the restoration of the original molecule.
Yeah.
Carbon dioxide.
Yeah, exactly. So we have to regenerate or restore, as you said, the original molecule that was used for the fixation, which is the ribulose 1,5-bisphosphate, right? So those are the 3 basic steps, okay? And in doing this, you ended up transferring this ATP to ADP and then diphosphate. But you use up the energy in that, and we donate electrons to hydrogen, so this NADPH becomes NADP. Okay, so let's see here. So, okay, the next thing is, this is just showing you the sort of what's going on, how everything sort of works together. In respiration, you have glucose plus oxygen, you get carbon dioxide and water and a whole bunch of ATP. So that occurs in mitochondria, both in plants as well as in animals. In photosynthesis, we have carbon dioxide and water, sun energy, and then you get glucose and oxygen. The waste products of cellular respiration are CO2 and H2O, and these are used in photosynthesis. The products of photosynthesis, carbohydrate and oxygen, are used in cellular respiration. Chloroplasts and mitochondria are the 2 organelles that specialize in converting energy into usable forms for cells involving 2 important biological processes: photosynthesis and cellular respiration. Okay, so that's just again a summary diagram. All right, so I think we can do some sample questions. So what I'm going to do is I put these in last night into Top Hat. It's tophat.com. I think you don't want to pay, but you can set up some of you may have set up already the free account. So I've uploaded this PowerPoint, so you can see all these questions. What we're doing here, they're all here, right? So, all right, let's go ahead and try these. Which of the following is an example of potential? And you see the joint code there. If you want to use it. Which of the following is an example of potential rather than kinetic energy? All right, so that's it. Let's take a look here. Okay, so the correct, so here's the choices. So water rushing over Niagara Falls, that's going to be kinetic energy, because of kinetic energy is energy of motion. So the water before it actually goes over Niagara Falls, that would represent potential energy, right? But when it's actually falling, that's kinetic energy. A gymnast sleeping, that's kinetic energy. Basically, the gymnasts have breakfast, hopefully, and they're taking chemical energy and using that within their muscles to do energy of motion. If they're actually leaping. A loaf of bread might have been, she might have had breakfast. So that would represent potential energy, because it's chemical energy can be thought of as a type of potential energy, right? And bread would have a lot of things like starch, which is a polymer of glucose. A crawling beetle foraging for food would be kinetic energy. Which term most precisely describes the cellular process of breaking down large molecules into smaller ones? Okay, I'm just gonna, there's a lot of questions to get to, so go ahead. All right, so the correct answer here is catabolism. So we're breaking down large molecules. A dehydration reaction is actually a type of synthesis reaction, so that would be an anabolic reaction. It's a synthetic reaction, right? Think Arnold Schwarzenegger, that's anabolic, he's building his muscles up, right? Okay, according to the induced hypothesis chemical of enzyme catalysis, the binding of the substrate changes the shape of the enzyme's active site, right? So B is correct. Some enzymes, but not for this. This is not the induced bit. Some enzymes do change their shape when activators bind to the enzyme. So that's different than the induced bit model. Typically, those activators are binding to a site that's other than the active site. The binding of the substrate depends on the shape of the active site. That's a true statement, but that's not what they're talking about here. The fact that the substrate, when it binds to the enzyme, actually changes the shape of the active site. That's correct. So you have to be a little bit careful when you answer these things. It may be a true statement, but it's not answering the question. All right, the chemical reaction that has a positive Gibbs free energy is best described as endergonic, right? Because remember, we did these like bar graphs. So if you're going up in terms of having more energy, it's going to be an endergonic reaction. So it's a reaction that requires energy. Okay, why is ATP an important molecule in metabolism? The basic idea has to do with energy coupling. It provides energy coupling between exergonic and endergonic reactions. So let me draw this on the board. So remember, we have something like glucose up here. It's a complex molecule with relatively equal sharing between carbon of electrons between carbon and hydrogen. When you burn glucose, you get CO2 and water, both of which are pretty stable electrons. So this is sort of a downhill reaction, right? It's an exergonic reaction. So we can actually generate ATP from this, right? That's what your cells do all the time, doing aerobic respiration. And then you're going to have other reactions. Let's say we have a synthesis reaction where we're building up. Let's say we take some glucose and we want to store it because we're a plant cell in starch and we want to make starch. Well, that takes energy. So this ATP can be used. So ATP will go in here as ATP. It'll come out, it'll be hydrolyzed, it'll become ADP plus inorganic phosphate. But we're helping to create this complex molecule called starch. So we're taking it, so ATP is the key element, coupling this exergonic reaction with an endergonic reaction. Okay. Which of the following is true of enzymes? So C is correct. An enzyme increases the rate of chemical reaction by lowering the activation energy barrier. So for all reactions, even exergonic reactions, there is an input of energy that has to happen. So this is our Gibbs free energy over here. And here's time. So let's say we have some reactants over here. And here's the products over here. So this is clearly an exergonic reaction because we're going to have a negative delta G, right? As we go down, it's going to go downhill. But the problem is we have to get over this barrier, right? So in a lot of chemical reactions, one way to get over that barrier is just to add thermal energy. So for example, you might do this in a chemistry lab where you have these products in a beaker, and then you heat the beaker up, right? And that supplies the energy to get over this bump. So this is the activation energy. But you don't have that luxury of doing that in your cell. If you just heat your cells up, you'll kill yourself, right? You can't just turn on a Bunsen burner and do that. So what your enzymes do is they'll lower this activation area energy. So the energy barrier for this reaction to go forward is much lower. And now just the regular thermal energy within your cells is enough at your internal core body temperature. So yeah, so actually, and this is some answer this, but enzyme function is dependent on physical and chemical environmental factors. So they probably read that wrong. All right, let's go to the next one. Both cellular respiration and fermentation have a common initial stage, which is Okay. Yeah, glycolysis, everyone got that. Go to the next one. Human muscle cells use lactic acid fermentation to okay, so what? Well, that's correct, actually. Okay. Okay, let's look at this carefully. So human muscle cells use lactic acid fermentation to produce more ATP than is possible through aerobic respiration. That's not possible, right? Aerobic respiration produces most. Produce ATP using electron transport chain. The lactic acid fermentation does not involve the electron transport chain, which is on the inner mitochondrial membrane. Lactic acid fermentation occurs in the cytoplasm. Regenerate NADH. So that's incorrect, right? Because what you're not, I actually was thinking of that one, but you want to regenerate NAD, right? You need to have NAD for glycolysis to go forward, right? So if that had said regenerate NAD, that would be correct, but regenerate NADH is incorrect. So it's a little tricky there. And then D is correct, produce ATP without oxygen, because you want glycolysis to be able to go forward. The only way it can go forward is to regenerate NAD, right? And then NAD can accept electrons. It ends up accepting electrons within glycolysis. All right, we'll go to the next one. Okay, so electron carriers, which electron carriers function in citric acid cycle? So it's NADH and FADH2, right? So NAD can accept 2 electrons, becoming NADH. FAD can accept 2 electrons, and it becomes FADH2. Obviously, it's also accepting, it's picking up 2 hydrogens. Okay, so during aerobic respiration, which of the following directly donates electrons to electron transport chain? So it's NADH and FADH2. Okay, during the electron transport chain, water is formed. Where does the oxygen atom for the formation of water come from? So there have been studies, because it's a good question, like where does it come from? One possibility is it comes from the fuel, such as glucose, right? When you have a meal, you're going to derive some glucose from it. And that certainly contains quite a bit of oxygen. But they did studies in which they used radioisotopes of oxygen. And we know now that the oxygen that's in water, that's formed through aerobic metabolism, actually comes from the atmosphere, right? That's the oxygen you breathe in, right? So this is why you need to breathe oxygen, right? And why you would die without it, because you're breathing in O2 as a gas. Okay, let's take a look at this one. Okay, so a cell has enough available ATP to meet its needs for about 30 seconds. What is likely to happen when an athlete exhausts his or her ATP supply? So that wouldn't be much fun to watch the Olympics if after 30 seconds, all the athletes had to sit down or completely exhausted. So that's not correct. So B is correct. Catabolic processes are activated in muscle cells that generate more ATP. So we have different catabolic processes. The first one is aerobic respiration, right? So as long as the athlete is able to breathe, they're able to do aerobic respiration. Now, for something really intense like a sprint, that breathing is not really sufficient. So you also have another process, which is the anaerobic metabolism, which we talked about, right? You have lactic acid fermentation, which is a form of anaerobic metabolism, which generates ATP without oxygen, right? So think someone who's doing weightlifting, judo, or any kind of martial arts where they have like sudden movements they have to do that involve lots of strength. That would be using a lot of anaerobic metabolism, such as lactic acid fermentation. And there's some other processes too, which we haven't talked about, but they're all catabolic processes that occur within muscle cells that generate more ATP quickly. Right? So that makes sports exciting to look at. D, other cells take over the muscle cells that have used up their ATP cease to function. That's not true. Let's go on. Okay, so most people got it right. When oxygen is released as a result of photosynthesis, it is a direct product of the splitting of water molecules, right? That occurs during the White reaction. Remember, water is the source of the electrons, right, that are fed into the photosystem 2. All right, during fall, the leaves of deciduous trees change colors to red, orange, yellow. This is because blank. What does deciduous mean? No one knows? Deciduous. What kind of a tree is a deciduous tree? Okay, it's a tree. Most trees lose their leaves. Those are deciduous trees, as opposed to evergreens, right, like pine trees. Let's take a look. Yeah, everyone got it. So what happens during the fall, and this is actually spectacular right about now, is that the chlorophyll is actually degenerated. It's essentially removed from the leaves. So it's no longer present in the leaves, but you have other pigments within leaves. They're there the whole time. Just they're sort of masked because of the green color. So you have these carotenoids, and there's some other ones also, but the carotenoids are the main ones. Pigments found within the leaves, and they're going to give you all these different colors: the yellows, the oranges, and the reds. There's actually another pigment that gives you the dark red too, but this is probably the main culprit. Gives us the colors. All right. Go on. Yeah, so the reaction that produces molecular action, oxygen, O2, takes place in the light reactions only. So that's the hydrolysis, where you hydrolysis means to split apart water. So that occurs fairly early on within the light reactions. Okay, what's the primary function of the Calvin cycle? It's to synthesize simple sugars from carbon dioxide. Right, because you actually start with a three carbon hydrocarbon, and then you have two of those, right? And then those are used to make glucose. From glucose, we can then quite easily make fructose. Glucose and fructose then make sucrose. All of those are considered simple sugars because they're either monosaccharides or disaccharides. Okay, what two molecules are the link between the light and dark reactions of photosynthesis? So ATP and NADPH, right? When you remember when we filled out your sheet, right? It's like the whole point with the light reaction, which is you were generating ATP, so it's chemical energy. You're also picking up electrons, and NADPH will carry those electrons. So that chemical energy and the electrons will be carried essentially to the Calvin cycle, and that's then used for synthesis reactions there. Yeah, so the wavelength of light absorbed by photosynthetic organisms, it's in the visible spectrum. So remember, we showed that, I showed you a graph where chlorophyll tends to pick up light in what range? Well, it likes blue light, and then it likes a bit of red light, and it doesn't like the yellow and green in the middle so much. And those end up being not absorbed, so they either go through the leaf or they're reflected out from the leaf. That gives us the green color. I think that might be it. You see, it says end here. Yeah, would that be on? Okay. All right, so that's it. That's all my questions. So you can review these questions also by looking at the PowerPoint that I uploaded, okay? All right, so I'll keep up. You got a question?
So you said, plants have to conform so they can spring like that.
Yeah.
Under what circumstances would, so when plants go to.
Oh, they have to do it all the time. So plant cells do make ATP directly, right? So you would think, well, why would they need to bother with all this other stuff? Well, ATP is not very convenient for storing energy. It's a fairly big molecule. So for example, humans, you couldn't, the amount of ATP you go through per day is like a huge stack of it, right? It's the same thing for plants. It's impractical to store everything as ATP. And so you've got to use glucose and polymers of glucose like starch to store energy. And then we're going to use that whenever the plant needs to grow. So it's building its walls, it's doing all sorts of things. It's the cellulose, right? So the glucose can be used for building cellular walls, things like that. Also broken down for energy for ATP, for various things. So basically, they're always doing aerobic respiration, just like our cells are pretty much always doing aerobic respiration. And there's also days like there's also times when the problem with solar energy is it's not always present, right? So at night, if you relied on just ATP being generated from photosynthesis during the daytime, you'd be out of luck during the night, right? So plants will continue to grow and everything at night. So, and they're always pumped. They have a lot of the same problems we have. They have to pump ions. They have to move things. Yeah.
I have a couple questions about the lab.
Yeah.
First one is, what is this? I was trying to figure out what promotes it and what.
Oh, okay. So enzyme specificity has to do with it. So for every substrate, you have an enzyme. So enzymes have very specific substrates they work on. It has to do with the specificity of the active site. The active site has a shape that will accept certain substrates.
Okay.
So it has that's why you spend a lot of time on the three-dimensional shape of the enzyme. Now there's a little bit of so say you have an enzyme that works on glucose. It might work on fructose because it's close enough, but not as well, right? Okay. But it would definitely not work on sucrose because that's a disaccharide, right? So that gives what would inhibit enzyme activity? Well, you have specific inhibitors that can sometimes bind to the active site. You also have enzyme inhibitors that can act allosterically. They'll bind to a site other than the active site, causing conformational change or shape change in the protein. I don't know if we really did that. We didn't play around with inhibitors or activators or something. Yeah, but those were, you can look up in your textbook, you'll find a lot of, or you can just Google it, you'll find lots of inhibitors of enzyme activity. Typically, they act on either an active site or some other site of the molecule.
I was confused on the wording.
Well, how do temperature and pH have similar effects? Oh, on enzyme activity.
Okay, okay,
Yeah.
okay, that makes sense, yeah, that's what I figured I was
Yeah,
Thank you.
okay.
Yeah, okay, I'll find it. What is this question asking?
Would you expect similar results if the substrate concentration were varied in the same manner as the enzyme concentration? What did you vary? Did you vary?
This is.
You varied the amount of
Yeah,
enzyme,
yeah.
right? Would you expect similar? Well, no, you get different results. See, here's the thing, it goes back to that curve. Let me stop this.